Biology Part 47. The maintenance of constant condition, often described as maintenance of constant internal environment, is called homeostasis. The liver is very important for homeostasis because it directly determines the concentrations of many different substances in the blood plasma. These activities collectively are called the regulatory functions of the liver, and they are described below. Let's talk about the number one, regulatory glucose. The body obtains glucose by digestion of disaccharides and polysaccharides in diet. In addition to glucose, and diet provides a mixture of other monosaccharides, but these are converted to glucose into the liver. The concentration of glucose in the blood is not absolutely constant but varies between 0.67 and 1.4 grams dm cubic, depending on the conditions. After a meal, the level rises, while after fasting, it becomes lower. Glucose is the only fuel taken up by the specialized cells of the brain and nervous system, and the minimum level of 0.6 g d cubic is just enough to keep these essential regions properly supplied. Glucose levels are kept within the normal limits in several. After a meal, the liver releases more glucose from the hepatic portal vein than it is required in the general circulation. Under these circumstances, glucose is converted to the polysaccharide storage material called the glycogen. The amount of glycogen is formed controlled by the hormone called insulin. Insulin is released from the pancreas and acts mainly by promoting the uptake of glucose by the membrane of liver and muscle cells. Up to 60 grams of the glucose can be stored as glycogen in the liver and up to 150 grams in the muscles. Above this limit, excess the glucose is converted into molecules of fat. Droplets of fat leave the liver via the hepatic vein and are taken up for storage of adipose cell. Mostly under the skin. Storing surplus energy in this way is a great advantage for a mobile animal because a fat contains more than twice as much energy as per gram as carbohydrates and proteins. Except in periods following a meal, the liver releases more glucose than it receives, breaking down its reserves of glycogen and also manufacturing new glucose from other organic compounds. While glycogen breakdowns, called glucogenolysis, is controlled by two hormones, glycogens from the pancreas to adrenaline and the adrenal glands. Both hormones are released in response to a fall in blood glucose, but adrenaline release can be also speeded up by the nervous system so that it boosts the availability of glucose at times of a sudden stress. The liver supply of glycogen is limited and must be supplemented by the manufacture of new glucose after only a few hours. The new glucose is made mostly from the lactic acid and released from the muscles and from the glycerol produced from the breakdown of fats. During prolonged fasting, amino acids are also used. The manufacture of new glucose is called gluconeogenesis, is promoted by glucagones, and up to 180 grams per day can be produced. The biochemical pathway used reverses the process of glycolysis and which glucose is broken down in respiration as described in the same tutorial.